Thanks, Mary. I of course, knew both Norman Kretchmer and Phil Sunshine. They'd both be really proud to have Paul Rozance here as the 28th Kretchmer lecturer. He's going to talk to us about new roles for old hormones. That has a longer title than that, but he'll give us some, I think, really interesting insights. Paul has ties to the Bay Area. He attended UC Berkeley. I forgive you for that, but as an undergraduate, but he left to attend Georgetown University Medical School in Washington and then returned to Stanford for his internship and residency. We tried to keep him here, but we lost him to the University of Colorado. Phil and I tried desperately to do that. We knew he was going to be outstanding person in our field, but he went there to train with really good friends, Fred Battaglia in particular, and I think there are many other distinguished people there as well, but I think it's really interesting and impressive that he's currently the Frederick C. Battaglia Chair in Neonatology Research and directs the Prematurity Research Center program in which he trained at the University of Colorado. He also was the program director for a NICHD-funded T32 training program, which is really impressive and has trained many really outstanding individuals who still emerged from that program. His own laboratory has been funded continuously from the NIH since 2009, which is really an accomplishment these days, and I think it's very impressive. Over the years, his research has broadened, and he's been studying the role of fetal-derived signals in regulating placental function and maternal metabolism. And he uses novel approaches in sheep primarily to investigate the role of specific gene products in placental function and liver and pancreatic development. He's investigating the response of pancreatic islet cells to a variety of perinatal insults, including genetic manipulations using both sheep and mouse models, the latter being used to determine long-term impact of perinatal insults on the developmental origins of diabetes and metabolic diseases. His basic physiological work has really been on his sheep, and that's a really good model for that. And I think that's in large part what's kept him at the University of Colorado. But he's been shifting his work towards mice, which gives me some hope. I've tried several times to recruit him back to Stanford, but I think the facilities there for sheep are really extraordinary, and what he has there with his distinguished colleagues is really a tremendous program, and we're just very proud of what he's been able to accomplish. Currently, as the president of the Society for Pediatric Research, is one of the most distinguished research societies in the country. With there are three parts of that. One is for neonatology, one is for obstetrics, the other is for basic scientists. And there's a limit to the number of people who can be in that group, and it's historically been there for quite some time. But Fred was a leader in that program, and now he's leading it, which again I think is really wonderful. So I don't want to take more of his time because I know he has a lot to tell us, but maybe I have a chance to recruit him back in the future, since now he uses mice, and we got plenty of mice at Stanford, so we have a chance. But anyway, welcome, Paul. It's really great to have you here.
Well, thank you so much, Dr. Stevenson. Thank you, Dr. Leonard and Schroeder, for the invitation, the whole organizing committee, really, and such a wonderful visit so far. I got to start it off with a nice dinner Sunday night, and then a wonderful day yesterday, and I'm looking forward to sharing research with you today and then concluding with a few more visits. But yeah, it's such an honor to be invited for this lectureship. I named in honor of Dr. Kretchmer. I never had the opportunity to know Dr. Kretchmer, but I wish I had. And I did have the opportunity to know Dr. Phil Sunshine, and I would love to, if you would give me the moment to just tell you and everybody, publicly how important he was to my career. I was only here as a pediatric resident, but that's actually when I first got to meet Dr. Sunshine. He took care of my child right here in the NICU, and my child was essentially born late preterm, and if you name the 5 or 6 things that complicate late preterm babies, Orion had pretty much every one of them. And that's when I first really met Dr. Sunshine, and he's definitely the only attending that my wife Brenda remembers from that time there. So he made a wonderful impression. Then I've gotten to pretty much kind of interact with them in a lot of various ways over the years. And so it's a true honor, and just acknowledging that his passing has, affected probably hundreds of people around the country, around the world. So the title of my talk today is New Roles for Old Hormones, and then more specifically, Lactogen, Glucagon, and the regulation of placental function and fetal growth. And I thought, how do I give you this sort of, one slide intro to the goals of my research? And as I mentioned, I never had a chance to meet Dr. Kretchmer, but in sort of preparation for this talk, I read more and more of his articles, and I don't think he's that well known as a placental physiologist, but I did come across this one article entitled Biological Factors Affecting Intrauterine Growth. And I would say if there's been, one thing that's sort of dominated a lot of the work I've done in the last 10 years or so, it's been looking for this fetal control of placental function and the regulation of fetal growth. Some of my mentors at Colorado, really pushed me in that direction, mostly Dr. Giacomo Meschia, who also recently passed away, I should mention. So that's been the overriding goal of my a major part of my laboratory in the last 10 years. But I came across this article, and I think it sums up sort of, that goal really nicely by Dr. Kretchmer. And I'm just going to read it because I really loved it. It's consideration of those animals with a placenta must involve a discussion of the components interrelated with the fetus, that is, the environment, the maternal component, the maternal placental relationship, the fetal placental dependency, and finally, consideration of the fetus as an active participant rather than as a parasite having a minor role in its well-being. So, this might have been he might have been contrary to a prevailing idea at the time, but it's certainly been something that, others, including Dr. Kretchmer, and people at my laboratory have thought about for a while. It's just we haven't really been able to nail down the exact pathways, especially as they relate to the regulation of placental function and fetal growth. So here's going to be my presentation to you of the sort of prevailing paradigm of the regulation of fetal growth. It's really a top-down phenomena, and I'm mostly focusing in on the regulation of growth in the latter half of pregnancy, which is when we see the disorders of growth pop up, things like placental insufficiency and intrauterine growth restriction or fetal overgrowth. So it does start with pregnancy, and in this context, it starts with the placenta. The placenta secretes these hormones into the parental circulation, into the uterine vein. These placental hormones change that environment. They change that, classically, that metabolic environment. They also change the cardiovascular environment, the cardiovascular adaptations to pregnancy they're somewhat responsible for. So what this does is it creates an environment in the pregnant person of a nutrient and hormonal situation that can support placental growth and fetal growth. And these nutrients, these hormones, are delivered to the placenta through the uterine artery. The placenta senses these hormones, senses these nutrients, takes those nutrients up, metabolizes them, yes, and then transfers them into the fetal circulation, where they're picked up by the fetal umbilical circulation and returned via the fetal umbilical vein to the fetus. These hormones then stimulate the production of insulin in the pancreatic beta cells, the production of IGF-1 in the liver, and these hormones then are anabolic growth factors, but they also regulate fetal metabolism. So you can see how fetal growth and metabolism are linked in this manner. However, I've already mentioned a few of the disorders of fetal growth, things like placental insufficiency and fetal growth restriction. We also have fetal overgrowth in the case of most typically parental diabetes. And the problem is that we don't know how to treat these as effectively as we should. In the case of fetal growth restriction, we really don't have any treatment. And even in the case of fetal overgrowth, we still don't fully understand that process. Okay, so let's talk a little bit more about fetal growth restriction. So fetal growth restriction can be defined as a failure of the fetus to reach its genetic growth potential. The most common cause is placental insufficiency, which also increases the risk of preterm birth. Statistical definitions such as small for gestational age are often used as a proxy for fetal growth restriction, or FGR. The complications of FGR are pervasive, both in terms of organ systems and lifespan. However, the precise incidence of FGR, and therefore the precise contribution of FGR to disease risk, is known, And I find this to be a really interesting topic, and I would refer you to this paper by Dr. Wilcox that goes into it more fully. I won't go into it. Finally, there are no effective treatments for FGR. And I'll show you an old study that makes the point and was really important in my work. But even as recently as the last 5 years or so, when we've tried something like sildenafil for the treatment of FGR, it didn't work. It actually created worse perinatal pathology, neonatal pathology than no treatment. So of course, this placental insufficiency causing fetal growth restriction leads to prematurity, and both of these things become even more important when you consider the fact that they lead to these lifelong increased risks for things like diabetes and other adult non-communicable diseases. And once again, I go to that article by Dr. Kretchmer, and he, even though this idea hadn't been as prevalent at the time, he sort of summarized what we think is the cause of this. And I'll read it again, or I'll read this quote. The major hazard imposed on an infant of either extremes of weight is the inability of the infant to adapt to environmental stress, and consequently, the infant is at greater risk for morbidity and mortality. I don't know if he was specifically referring to adult morbidity and mortality, but it really is that inability to adapt to subsequent environmental stress that's key. So now I'm going to talk to you about this study that was one of the first studies I read as a fellow. And it's really been with me, ever since, and I'll explain why. So this was a study in which they randomized women who were at risk for having fetal growth restriction or intrauterine growth restriction. They randomized them in the middle of pregnancy, so you can see by 20 weeks, and they took these women who were at high risk for having intrauterine growth restricted pregnancies or fetal growth restriction, and randomized them to 3 different treatment groups. The test and the hope was that this group that received 40 grams of protein in this daily nutritional supplement would have less fetal growth restriction than the control group. They had that group. They had a group that had the same number of calories, but only 6 grams of protein. And then they had a control group, which had the same looking item, but it was mostly vitamins and not really even calories. And what they found was, as you can see on the left, that the group that got the high protein supplement, despite all the animal evidence that said this should have worked, actually had a higher perinatal death rate, both before birth and immediately after birth. And then in the analysis after this study, Dr. Rush looked at the percent of calories in different studies. He looked at the percent of calories and nutrient supplementations that were provided as protein and found an association with lower fetal weight at the end of that trial. And these changes in weight aren't really that big, but I think the signal was there that maybe something bad was going on, which was obviously confirmed in his big randomized trial. So this really bothered me because I couldn't understand, given the paradigm, why that should have happened. I could understand why it wouldn't work. I could understand that that if you gave excess nutrients to a person who has placental insufficiency, maybe the placenta is not going to work. It's not going to transfer those nutrients as well into the fetal circulation. But why should there actually be increased mortality? It didn't make sense. And that's why I come back to this idea that the model is incomplete and there are these clinical implications, as I mentioned. So as Dr. Kretchmer pointed out, and as a lot of other people have pointed out, it's long been postulated that the fetus ought to have signals to the placenta to impact placental function as it relates to nutrient transfer and the regulation of growth. However, we've never really been able to identify those signals. And so just to put this into a cartoon, we have a lot of physiological pathways that have been worked out where the parental environment controls placental development, function, and nutrient transfer to the fetus. We have obviously the hormonal pathways and other pathways where the placenta regulates maternal or parental metabolism and cardiovascular adaptations to pregnancy. And we have the regulation of the placental regulation of fetal growth by nutrient transfer and other mechanisms. However, what's postulated but not known, or not until recently, is how does the fetus actually regulate the placenta? And even more, can the fetus regulate the maternal environment through the placenta? Okay, so here's that pathway I talked about. The other thing that I haven't mentioned yet is what Dr. Mesquia said to us, me and my colleagues, was not just that we should find a regulator of a fetal regulator of placental function. He said there had to be a fetal inhibitor of placental function. And I think that's really important. And in fact, that's what we've shown. And that's what I will show you. Not only is there a fetal inhibitor of placental function, but there's also this pathway inhibits placental secretion of hormones, changes potentially the maternal environment, as well as even the cardiovascular adaptations to pregnancy. And the placental hormone is lactogen. The fetal hormone is glucagon, and what regulates glucagon secretion is hypoxia and amino acids. And while I won't talk about it right now, I think there's really important physiological implications for an inhibitor of placental function, especially as it relates to amino acid function. There's evolutionary importance, and then this whole idea of the need to really match placental size and function to fetal growth potential. Okay, let me talk to you a little bit about glucagon physiology. Glucagon secretion is by the pancreatic alpha cell and is classically described as central to a homeostatic feedback loop which maintains glucose concentrations. Glucagon also is central to a homeostatic feedback loop which maintains amino acid concentrations. Fetal glucagon is increased with fetal acidosis, hypoxia, and fetal growth restriction. Despite these observations in human fetuses, glucagon was previously thought to have a really minimal role in fetal metabolism. However, we've recently demonstrated that this amino acid regulatory homeostatic feedback loop that with glucagon is present in the fetal sheep. And here's a couple of studies to make that point. When we infuse amino acids into the fetal sheep, just like in the human, we get an increase in fetal glucagon concentrations. And then when we infuse glucagon acutely into the fetal sheep, we get this immediate lowering of fetal amino acid concentrations. Additionally, fetal sheep glucagon secretion is also stimulated by catecholamines and hypoxemia, which we've shown, and fetal sheep glucagon is increased in experimental placental insufficiency and fetal growth restriction. So I think this is a good time to stop and talk to you really quickly about why we use pregnant sheep to model human pregnancies and why it's so important for placental and fetal physiology. Essentially, the reason why it's been used as a model for these processes for almost 100 years now is the fact that you can do surgery on the fetus to place indwelling catheters in the fetal circulation. You can also place the indwelling catheters in the maternal circulation. You can then sew the animal back up. You can have the catheters exposed through her pouch on her flank, and she can recover from anesthesia. And you can maintain these catheters for weeks or months. And with that, you can then sample blood that represents the uterine artery, the uterine vein, the umbilical artery, the umbilical vein. We can also trace water, which tells us how fast the blood is flowing, both in the umbilical fetal circulation as well as the uterine circulation. And with the knowledge of concentration differences on either side of the placenta and blood flow rate, we can calculate a utilization rate by the placenta or the fetus, and we can even take the difference, and that tells us the placental metabolic rate for a substance. Add into that the fact that we can trace things like glucose and leucine, and we can get these really detailed fetal nutrient metabolic tracer studies. So that's why the sheep is, remains a really relevant model for the study of placental and fetal physiology. And what about our sheep model of placental insufficiency and fetal growth restriction? We have a very robust one. It was one that was created based on the observation and made by shepherds in Australia that the sheep that were gestating during the hot months had smaller fetuses than the sheep that were gestating during those same months, but at higher latitudes where the lower latitudes where the temperature extremes weren't so high. And this ended up being harnessed into an experimental model of placental insufficiency, where we take sheep whose pregnancy is 148 days and expose them to elevated ambient temperatures starting around day 35 and leaving them in those elevated ambient temperatures until about 105 days gestation. When you remove those animals, they have smaller placentas. And in fact, whether they remain in the high temperature or not, we see this progressive decline in uterine and umbilical blood flows, a decrease in placental weight, decrease in placental oxygen, glucose, and amino acid transfer to the fetus, decreased fetal concentrations of oxygen, glucose, as well as insulin and IGF-1 concentrations. And we see high fetal glucagon concentrations and lower maternal placental lactogen concentrations. So these are all things we see in cases of human placental insufficiency. What I'm going to tell you about this negative regulator of placental function is not to suggest that the classic paradigm is wrong or is not of really central importance. And to highlight that, I'm going to show you data from our model of IUGR. This is a bit of a meta-analysis. It represents about 100 animals that were all studied by myself or two of my close colleagues in essentially the same way. And we decided to pool the data really to look at sex differences for a grant that I was going to submit. And we realized there were no sex differences in the things we were looking at, but we thought we had a really important story to tell just by pooling all the data from 10 or 15 years or so. So you can see on the top that both insulin and IGF-1, I mean, in the case of insulin, it really discriminates the two groups fairly well. The IUGR groups are all have pretty low insulin concentrations. IGF-1 on the top graph doesn't discriminate quite as well, but of course they both track and associate with fetal weight really nicely, IGF-1 more so than insulin. So here's glucagon. Glucagon we measured a lot less frequently back when I first started. And this shows you that, yes, with a lot of animals, we can detect a difference between control and IUGR fetus and their glucagon concentrations. But that discriminatory power is really pretty weak if you ask me. And the association also is there because we have so many animals, but it's not the strongest association. But that's essentially, what we're showing is just this association. So we went ahead and looked at the relationship of fetal glucagon concentrations to some of those important fetal metabolic, placental physiological functions like uterine blood flow, placental oxygen uptake, and placental leucine uptake as a marker for amino acids. And you can see that the association is there that you would expect for an inhibitor of placental function. Animals with higher glucagon concentrations tended to have lower rates of all of these processes. But those are just associations. So our next step was to move forward with the experimental testing of this idea that glucagon regulates or inhibits placental function. So we used our sheep in the way that I just described. We performed surgery at about 117 days gestational age to place those catheters that I talked about. And then after the animals recovered, in about 5 to 7 days, we began our experimental infusions. One group got glucagon directly into the fetus. The other group got our vehicle. They were our control group. And at the end of that, we measured uterine and fetal umbilical blood flows and nutrient uptakes. We did leucine and glucose metabolic tracer studies, which I won't show you the data of today, but this has been published. So I'm going to summarize in one slide, pretty much one slide, what we found. And here you go. So while I said that we set this experiment up to look at placental function, it was quite a surprise to see a drop in uterine blood flow. This is from a glucagon infused to the fetus. The glucagon does not cross the placenta in either direction. So this is truly a fetal hormone decreasing parental, or in this case, maternal uterine blood flow. We also found lower placental amino acid uptakes, lower placental glucose uptakes, and lower placental oxygen uptakes. We saw a reduction in placental glucose and oxygen metabolic rates or utilization rates. We saw a decrease in fetal nutrient uptakes. Most profoundly, it was the amino acids that decreased the most. So their concentrations in the fetal circulation were low. As we might expect then, the low amino acid concentrations we thought were what led to lower beta cell stimulation. So these animals had less beta cells, their pancreatic beta cells secreted less insulin, they produced less insulin, and they had a lower replication rate. And with this, both insulin and IGF-1 were reduced, and fetal growth was small. So after just 9 days of a fetal glucagon infusion, we found all these things, including animals that were fetuses, I should say, that were about 15% lower in terms of their weight. But what was a really unexpected finding, and in some ways a serendipitous observation that we made, was that the maternal placental lactogen concentrations were reduced after these 9 days. And those are the data that I'm going to show you from this study, the individual data, because they were so impressive. So here you can see the maternal arterial plasma placental lactogen concentrations. We can actually calculate using what I described earlier. It's called the Fick principle, since we knew the blood flow rate and we could measure placental lactogen on either side of the maternal placental circulation. We could actually calculate a secretion rate, and we found this profound reduction in placental lactogen secretion. And while this was actually our first indication that something was going on, with placental lactogen, it was this placental lactogen mRNA expression, which was 50% reduced. It was, it becomes noteworthy in a little bit because it starts to get at our future studies as to how glucagon is doing this. So these were some of the most profound changes that, you know, myself and my colleagues had really ever seen with sheep studies. And by this point, I was working with a great collaborator, and I still am, Dr. Russ Anthony at Colorado State, and he has spent his, you know, decades-long career studying placental lactogen. That's why we had the RIA to measure the concentrations in the circulation. It's why we even ran the real-time assay for the mRNA concentrations in the first place. And he had never found anything that could regulate placental lactogen to a degree like this, let alone inhibit it. Okay, so now it's going to be time for me to talk to you a little bit about placental lactogen, which is the other hormone that I'm talking about today. This is a cartoon of you know, the placenta. You've got on the top the fetal side of the placenta, on the bottom the parental side of the placenta, and you know, you can see the blood vessels running into the fetal villi. The fetal villi in this cartoon are surrounded by a layer of cytotrophoblasts. These are mononuclear or single nuclear trophoblast cells. These single nuclear trophoblast cells end up fusing, and early on they fuse and create a syncytial layer that's on the top. It's the syncytial trophoblast, and throughout pregnancy they continually fuse with that syncytial layer above. And it's really thought to be that syncytial layer that produces a lot of the placental hormones that are secreted into the maternal circulation. It's the cell, or it's the syncytium that's responsible for the nutrient transport. That's the classic thinking. And those villi and that syncytial trophoblast is in direct contact with the maternal circulation. And on the bottom, you can see those blood vessels that are being remodeled. They're remodeled by these trophoblast cells that actually get out of the villus and migrate, and some actually get into the wall of the vessel so that you get this dilation. And so these villi with their syncytial trophoblast lining are bathed in maternal blood. Okay, so what's that? Let's talk about placental lactogen now that I've oriented you to a little bit of anatomy. So placental lactogen, it's a peptide hormone belonging to the growth hormone and prolactin family. It's made primarily in those syncytial trophoblast cells in the placenta. It's the most abundantly produced hormone by the placenta. And circulating concentrations were historically considered as a way to identify pregnancies at risk for intrauterine fetal demise or even potentially fetal growth restriction. How does it work? So here's the classic thinking behind how placental lactogen works and say promotes fetal growth. So placental lactogen is secreted from those syncytial trophoblasts. What I didn't point out is that it's actually secreted into the fetal circulation as well, which certainly calls into question directionality of the secretion by the syncytial trophoblast, or even if it's secreted by any other potential cell there. But what its most recognized role is to go into the parental circulation, and there it promotes IGF production. It promotes or it creates a state of peripheral insulin resistance, and then of course it, as its name implies, it prepares that pregnant person for lactogenesis after delivery. Well, especially the top two, the IGF-1 production and the promotion of peripheral insulin resistance, those do a few things. One is they create that glucose intolerance, which frees up glucose into the circulation of the parent. Promotes lipolysis and ketogenesis to create a fatty acid pool that's now circulating and available for the placenta and fetus. And to some extent, it may promote proteolysis, freeing up some amino acids to get to the placenta. The placenta then transfers these. They're the sort of primary building blocks of well, both for energy as well as for accretion of new tissue in the fetus. So for fetal growth, they do those things that I described in the early cartoon by stimulating fetal anabolic growth factors. But in addition, placental lactogen itself can directly stimulate the fetal liver to produce IGF-1, as well as promote amino acid uptake and even maybe glycogenesis. This is just a couple of slides showing how, especially in the 70s, placental lactogen was considered potentially as a marker for being small for gestational age, growth restriction. And that holds then for humans in the graph above, as well as in a model of fetal growth restriction, as you can see below. I don't think it ended up having a lot of clinical utility after the 70s. We had better ways of looking for fetal growth restriction and placental insufficiency. but it certainly was there. And even in a really recent study, very severe pregnancies, severely FGR pregnancies at high risk for an intrauterine demise. This is work by Anna David out of the UK. She showed that in those babies, that those pregnancies where the severity of FGR was really profound in all subjects, those with the higher placental lactogen concentrations had a higher rate of an IUFD. So this has been confirmed in a study in 2020 as well. Okay, before this was the classic paradigm that I showed you before some of the work that I did with Dr. Russ Anthony. Nobody had really thought that placental lactogen would have a role in uterine blood flow, nor would it have a role in placental nutrient transport directly. It had the indirect roles by liberating these nutrients in the parental circulation. But if you think about it, if placental lactogen's goal or overall role is to promote fetal growth, you might predict that if it had an impact on uterine blood flow, it would be to increase uterine blood flow. And if it had a role directly on regulating placental nutrient transport, it would be to promote placental nutrient transport. So it'd be predicted to increase both of these. Now, rather than run the experiment to show that directly by giving exogenous placental lactogen into, let's say, pregnant sheep for the course of their pregnancy, we took a different approach, and this is really Dr. Anthony's work to technically work out this system and then apply it to his favorite peptide in the world, placental lactogen. And that is the impact. And so what I'm going to show you is the impact of experimental knockdown of placental lactogen in sheep. And the way we do this, which I didn't describe earlier when I talked about sheep as a model, is what we do is we get pregnant sheep that are about 8 days pregnant. We can retrieve their blastocyst with a surgery. Once you have the blastocyst, you can manipulate it in vitro and incubate it it takes about 2 days all told, but you incubate it with a lentiviral vector that you can then package in, something to deliver into the trophectoderm, which is the outer cell layer of the blastocyst. This trophectoderm is what becomes the placenta. And in this case, what we targeted there was an interfering RNA for placental lactogen. So this decreased placental lactogen expression really throughout pregnancy, because what you do is you take that blastocyst now and put it back into a recipient pregnant sheep, and they gestate just fine. Those that have the knockdown are smaller. And so based on his initial observations of those small fetuses, we went ahead and collaborated. He's up at Colorado State University. Those animals were produced there, and then we brought them down to my laboratory to do some of this physiological studies that I've already talked about. And here's what we found. We found a decrease in uterine blood flow, a decrease in placental amino acid, glucose, and oxygen uptake, a decrease in placental glucose oxygen metabolism, a decrease in fetal nutrient uptake, including amino acids. We think the lower nutrient uptake led to lower insulin and IGF-1, which we've shown, as well as lower fetal growth. So if this looks familiar, it should look familiar. That's the reason I'm showing it to you. That's the reason I have it in the exact same scheme. So the idea that, placental lactogen with that glucagon infusion is down, and we saw all those effects on placental physiology and fetal growth. Now we have some direct evidence that impairing placental lactogen production throughout pregnancy can do the same thing. So I'm going to go back to this 9-day infusion and look at this decrease in placental lactogen and start talking to you a little bit more about that. So first, the idea that glucagon could regulate placental lactogen. I mean, at this point, when we first started doing these studies, the only evidence that the glucagon receptor was even in the placenta was some work that was done partially by our chair of OBGYN, Dr. Santoro, and her colleagues back in New York when she was there. But they essentially were sort of manipulating glucagon receptor gene expression in different compartments of mice. And in this case, because of our involvement of Dr. Santoro, they did it in the placenta. So the only evidence was that when you did a global knockdown, or knockout, I should say, of glucagon receptor, you could look at the placenta and you could see that by real time, it really wasn't there. And by standard real-time PCR in the controls, it was there. So that was it. Never really been characterized or localized before. So I'm showing you some sheep placental slides that use RNA-scope to look at the message for the glucagon receptor, and then some immunohistochemistry to look at the presence of the protein. And in the sheep placenta, for those that aren't familiar with sheep placenta but are with human, you'll notice there's some clear histological differences there. But what I'm showing you is that the glucagon receptor really is in that layer of the syncytium. It's the syncytial cells. It's the interface between the fetal and the maternal parts of the sheep placenta where that glucagon receptor is present. I'm going to take a bit of show you some data not from sheep anymore. So we got to the point where we were really wanting to understand how glucagon was regulating placental lactogen. And for a variety of reasons, I think some developmental trophoblast biology reasons, it's hard to isolate trophoblast cells from the sheep placenta and get them to syncytialize. There's, as opposed to, maybe what's in the human, where most of the cytotrophoblast or a decent population of them can fuse and become syncytial cells. In the sheep, it's this really unique binucleate cell. So the cytotrophoblast that has one nucleus divides its nucleus, but not its cytoplasm. It becomes a binucleate cell. That cell is what fuses. It's really hard to get either the single nuclear cytotrophoblast to undergo that process and become syncytialized, or isolate the binucleate cells themselves and get them to sort of syncytialize in vitro. So we were faced with a challenge. And this is where really in human pregnancy research, the manipulation of trophoblasts in vitro after isolation is much better than it is in the sheep, in the animal model we were using. But we didn't know if the glucagon receptor was even there. So these were some of our first studies. Above is using RNA-scope again to show you that now in this human villi, which you can see the syncytium on the top, which is sort of facing what would be the intervillous space with maternal blood. You can see that we have the mRNA for the glucagon receptor there. And then with the other person I really started collaborating with on these projects, Dr. Teresa Powell, in her laboratory, she has the technique of taking sort of biopsied, even frozen placental tissue and being able to fractionate it in a way that you enrich for either the maternal-facing microvillus membrane or the fetal-facing basal membrane. And she's done that before. She's done that looking with her collaborator, Dr. Thomas Jansen, looking for things like the insulin receptor, the IGF-1 receptor, the adiponectin receptor. And most of the time, those things are localizing to the maternal-facing microvillus membrane, which really fits with that idea that the maternal hormonal environment regulates placental function. But in this case, this is not what we found for the glucagon receptor. We actually found that it localizes much more strongly to the fetal-facing basal membrane. And those are the Western blots below to show you that. We have our sort of positive control of the pancreas, the microvillus membrane then with very little glucagon receptor expression, and then the fetal-facing basal membrane where it seems to localize mostly. And we've done that for a number of subjects, and it seems to be a consistent finding. So having then established that the glucagon receptor is present in the human villi, in the human syncytial trophoblast even, we next wanted to carry out some of those studies to begin to, one, understand how this happens, but of course, to make sure that it does happen in humans, because we want our work in sheep to be relevant for the human pregnancy and the human conditions. So we ended up using two different cellular models of human trophoblasts, of in vivo human trophoblasts, and one are called vivo choriocarcinoma cells. And for those that don't work with these cells, I should tell you they are derived from a choriocarcinoma or a trophoblast tumor. They're now an immortalized cell line they're widely used in the field, although I would say widely maligned if you put a grant in, let's say, that uses only vivo cells. So they have the limitations that all immortalized cell lines do. There's some other immortalized placental cell lines, but these vivo cells are felt to be the ones to use if you're going to study syncytialization and the syncytium, because of the phenomena that you can see on the top. If you treat these cells with forskolin, forskolin is a cyclic AMP mimetic. If you treat them with forskolin, you'll push them towards syncytialization. So you get these multinuclear syncytial cells in vitro, and you can then study them for a small period of time. You can also, and we did that, we incubated them with glucagon for 24 hours after pushing them towards syncytialization for 48 hours with forskolin. The other, potential way you can study them is just as mononuclear cells, where you can we treated them with glucagon just for the 24 hours as sort of cytotrophoblast-like cells. The second cell that we use to model the in vivo condition in human pregnancy is the actual primary human trophoblast cell, which is isolated from term placentas. And in our case, we are just taking healthy pregnancies at term and isolating those primary human trophoblast cells. The benefit of these cells is that you don't need something like forskolin. You don't need a cyclic AMP mimetic to cause them to syncytialize. In pretty standard conditions, you will get them they will just syncytialize spontaneously. And this becomes important, especially for those of you who are thinking to yourselves, well, glucagon signaling is really through cyclic AMP, and now you're adding forskolin to the vivo cells to also stimulate cyclic AMP. You can see where it kind of gets messy. The other part that's messy about this is with the primary human trophoblast cells, I've already indicated to you that the glucagon receptor is on the fetal-facing basal membrane. And in the culture conditions, in 2D culture conditions, it's thought that these trophoblast cells, their basal membrane is going to be on the bottom. It's going to be adjacent to the glass or the plastic, whatever the container is that you're using. And it's the apical side that's available to media. So I'm telling you that because it took us a long time and a lot of frustration, a lot of concern, because we kept getting results that didn't really match our hypothesis, which is fine. It could have been that glucagon didn't do anything to these primary human trophoblast cells, but it wasn't until we started culturing them in these transwell systems, which is cartooned off to the right, that we saw an effect of glucagon. So I do think that there remains some localization of the glucagon receptor, even in these isolated primary human trophoblast cells. Okay, so let me just show you the results. As I mentioned, there were, I'll say, there were so many challenges with these cell studies. And as a person who, mostly studies animal physiology and isolated pancreatic islets, and I know exactly what to do and what to expect, it was challenging for these vivo cells and these primary human trophoblast cells for me to get the conditions just right, where we felt comfortable, where I could share the data with you. So what turns out to be a lot of work is going to be summarized, with two main slides. The first slide is using the vivo cells, and in that situation, what you can see is if we just take those mononuclear vivo cells and treat them with glucagon, and we can use everything from high physiologic to very high pharmacologic dosing for the vivo cells, we can see that with all doses, we get this inhibition of placental lactogen secretion, which is there graphed over time. We serially sample the media from the same set of cells. What has been really one of the things that caused us a lot of consternation is the fact that once we syncytialize these vivo cells, they really stop secreting placental lactogen. And it could be the forskolin, right? A forskolin, just like glucagon, might inhibit placental lactogen secretion, but it was doing the same with the marker HCG, which is the human pregnancy test hormone, the chorionic gonadotropin. So we did a lot of recovery. We did a 2-hour recovery. We did a 24, 48-hour recovery. And what happens is we definitely see a recovery from that forskolin treatment where lactogen secretion and HCG secretion go up like we'd expect. The problem is now our cells, by the time we hit them with glucagon, are sort of 6 days post forskolin, and they start to look very odd. They don't look like a cell where I'd be comfortable sharing that data with you all. But the important thing here for this slide is that forskolin, at least in the mononuclear vivo cells, inhibited placental lactogen, just like it did in the sheep. So more importantly, I think what happens in the primary human trophoblast cell, and again, after a lot of work using two-dimensional culture plates and other ideas around how to get access to the glucagon receptor, we ended up showing essentially the same thing. Now we only have 2 doses because these cells are a little bit harder to come by, but you can see that either, and these are some of the higher doses, at either of the higher doses of glucagon, we end up with this inhibition of placental lactogen secretion at 66 and 90 hours. There is this wave of pretty high placental lactogen secretion at 18 hours when we first sample the cells. So again, there is something about that single nuclear trophoblast cell that maybe is secreting some placental lactogen, maybe to the fetal circulation in vivo. The 66-hour and the 90-hour data are messy there. There's, especially in the controls, there's a ton of variability. I think that's prep to prep variability. And if just to make that point for you, we ended up normalizing both at 66 and 90 hours. We normalized secretion with glucagon to secretion without, just to show you that in the case of that variability, really everything is inhibited, with the exception of that one point in the middle. And then the mRNA concentrations, just like in the sheep, we find that glucagon inhibits placental lactogen mRNA in these isolated BWA cells. And importantly, this, I found this fantastic, to be honest. Glucagon does not inhibit HCG. So if this was sort of this, to me, it's better that it's specific for placental lactogen, and at least chorionic gonadotropin is not impacted by glucagon. So these cells are secreting HCG just like you would expect them to. So, I really like this quote by Dr. Kretchmer, and it's different from the first one, but I think it comes later in the paper, and it has it had a word I had to look up, proaction, but to make sure it was a word, but indeed it is a word. He used it in this paper, but I like to sort of, point out that there's this he writes, there's a harmonious relationship exists between external environment, maternal physiology, and homeostasis, placental integrity, including metabolism and transfer, and the fetal physiologic reaction and proaction. So, and I just love that idea of reaction and proaction, because, when I started in this field, it was all about fetal reaction. People thought there ought to be some sort of fetal proaction to regulate placental function. Dr. Giacomo Meschia was convinced of it and spent, the last 4 years while he was with us in the lab, really pushing us to find that fetal growth inhibitor. He was just convinced that was there. But like I said, he wasn't the only one. Dr. Kretchmer wrote about it right here. So I wish I could get up here and then go on to tell you about the clinical trials we've done to do risk prediction using placental lactogen, to tell you how we can manipulate this system, even in the cases of pregnancies with diabetes, so that, we will no longer have shoulder dystocia, fetal overgrowth, IUFD, even in the case of well-controlled parental glucose concentrations. I can't do that. This is really kind of where we are. But I do want to come back to this idea, this problem presented in the paper by Dr. Rush that really, honestly, I can't tell you enough how when I read this, all I wanted to do was write grants to basically show Dr. Rush's graph and say, please give me money to figure out a way that this might have happened. I couldn't do that, but at least now, we can at least propose a potential explanation for why this happened in his study. So I've already shown you that in regular, normally growing fetal sheep, amino acids stimulate glucagon secretion and raise glucagon concentrations. So these are from some other papers that we had done where we measured glucagon concentrations. And on the left, you can see that after 90 minutes, just like in the control fetuses that I already showed you, glucagon concentrations double. But if we sustain that, if we keep that amino acid infusion going, not just 90 minutes, but for a full week and a half or 9 to 10 days, the glucagon concentrations don't double. They go up 300 or 400%, maybe 300%. You can see that it's sustained. And I will point out that with something like fetal hypoglycemia, you don't actually get this sustained rise in glucagon. You get a transient rise, it goes up, and then it comes right back down after about a day. But at least for amino acids, it's a sustained rise in glucagon. So how does that relate to Dr. Rush's trial? Well, the subjects in that trial, the women, as they were described in that paper from 1980, they were already at high risk for having placental insufficiency and fetal growth restriction. So you can imagine that maybe there is some underlying fetal hypoxia in some of the subjects. Maybe that is stimulating the alpha cell to secrete some glucagon. So, not in everybody, but maybe in a few. Maybe placental lactogen was already decreased. And of course, as I haven't sort of told you, one of the hallmarks of placental insufficiency is a drop in uterine blood flow. So maybe they already were having a propensity for lower uterine blood flow. And now what you do is you start giving that person, in this case, the mother, every day you give her these exogenous dietary protein loads, which become free amino acids. That raises your amino acid concentrations. Those get across the placenta, even maybe despite some placental insufficiency, and they raise fetal amino acid concentrations, further stimulating the alpha cell to secrete more glucagon, which then over time inhibits further placental amino acid transfer. Well, that would be appropriate. That would be a homeostatic response, but it might also impact things like placental glucose and oxygen metabolism. It might further decrease placental lactogen secretion and even uterine artery blood flow. And that might explain, some of the results that he found. So I'm just going to summarize some of the things I showed you and then just give you a real quick taste of where we're going. Already sort of established was this homeostatic amino acid regulatory feedback loop that you can see in the bottom right. What I've shown you today, what that adds is that glucagon, in addition to regulating liver hepatic amino acid catabolism in the fetus, also inhibits placental function. It inhibits placental nutrient transfer, which then decreases fetal amino acid concentrations, leading to an inhibition of fetal growth. That also inhibits placental lactogen secretion, which may impact uterine artery blood flow. And this brings up a few testable points in the model that we've gone forward with. One being, the question of whether insulin and IGF-1 counterbalance glucagon like they do in the liver and other organs. The other being placental lactogen. If we give that back, can we actually promote uterine artery blood flow? Or does it cause uterine artery vasodilation and increase in blood flow? And the third being, if we give that fetus that's getting glucagon amino acids, we ought to be able to prevent the decrement in fetal growth restriction. So the first one I want to show you are the placental fetal umbilical amino acid uptake. So the transfer of amino acids into the fetal circulation. This is from that glucagon infusion paper, where you can see now individual essential amino acids were all down. You can see that insulin and IGF-1 actually do the opposite for some of these amino acids, especially those branched chain amino acids, isoleucine, leucine, and valine. And this is work by Dr. Jane Stremming. She's a fantastic early career faculty in our group working at our research center, and these were really her studies. And we had tested insulin and IGF-1 individually and thought, well, they didn't do this. They didn't promote placental nutrient transport. So some of us, I won't actually name who, thought there was a very low likelihood that these findings would be as they are. But to my surprise, here they are, which I think is important. So we have this, everybody's familiar with the insulin, the IGF-1 insulin axis counterbalancing glucagon action in the liver. Here it might be doing so in the placenta. Well, that was the good study. Here's the data that I'm not happy about, but it's fine to end this way. We predicted that placental lactogen would actually be a vasodilator. So we went ahead and tested it as an acute vasodilator and found absolutely no impact of acute maternal infusions of placental lactogen on uterine blood flow. So clearly that story is much more complex. And then the one that really has bothered me, and these are data just from even the last few months that I have to figure out, but I'm happy to share with you. I was convinced that amino acids would actually prevent the decrement in fetal weight, and they did no such thing. So clearly there's a lot more in the pathway regulating growth and metabolism that we have to work out, but we're looking forward to doing that. Here's some of the physiological implications. Here are the three people I should really thank. A postdoctoral fellow on the left, Amelia Tan, worked with Russ Anthony as her graduate advisor. I was on that committee as well, and I think she joined our group to keep working on this. Marge Wynn is now a new bioengineering PhD student who's staying in our lab, fortunately, and going to work on sort of the syncytial cytotrophoblast differentiation to syncytial trophoblast, things like scaffolding and organoids and such. And then Sarah Silvick's now a assistant professor at Wake Forest. She has her own R01, not studying this, but studying reproductive health and aging using non-human primates out at Wake Forest. There's my wonderful group at the Perinatal Research Center and everybody up here is integral to these studies. And then finally, I'll go back to this quote, which you have seen before, and that's consideration of the fetus as an active participant rather than as a parasite having a minor role in its well-being. And then when Sarah's study came out, Dr. Gatford was kind enough to write this sort of commentary on the study, and she described it as backseat driver. The fetus is not a passive passenger. So I hope we've been able to, put a name and put some names and more specificity to that pathway that Dr. Kretchmer, suggested in that article. So thank you very much. I'll turn it over to you for the rest of the time.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.